membrane space, and relatively more negative here. And what do opposite charges want to do? They're attracted. So these pluses want to get over on this side in the worst way for two reasons. One, by diffusion from high to low, right? And two, because opposite charges attract. So there's a really big drive to get in. And so in this way, what we have is this production as the electrons travel down. A proton gradient is formed. We call this the proton motive force. And this proton gradient is represented by a relatively high concentration of H+ in the intermembrane space. So you could picture the H+ in the intermembrane space. It's kind of like water behind a hydroelectric dam. You've got a dam, all this water behind the dam. What does that water want to do? It wants to go to the ocean, doesn't it? It wants to push through the dam, go through the turbines, turn the turbines and make electricity. Well, again, I'm going to try to use this, maybe I'll try to use this 24/8 one more time. So this is 24/8, and I'll pass it all the way through. This is an important protein, a motor protein here, called ATP synthase. If you have your text, you can open that up. So this is where we convert the proton gradient into ATP. This is huge. So as the only place where those protons can flow back into the matrix, if we go from H+ high to little H+ low, the gate is the ATP synthase. So the proton gradient is formed with a high H+ in the intermembrane space, and the protons can flow through ATP synthase back into the matrix of the mitochondria, the center of the mitochondria. Now what happens when protons flow through the ATP synthase? What does this name of this enzyme mean to you? It's an ATP synthesizing enzyme. It makes ATP. This is one of the most amazing proteins that we have. And as I said, when H+ flows through the ATP synthase, it turns a turbine in the protein, just like a turbine in a hydroelectric dam. It literally is a motor protein. And that mechanical kinetic energy that's generated is used to make ATP. So, protons flow through ATP synthase back into the matrix and produce ATP. And they produce ATP in this step by a process called chemiosmosis. And one of the things that we want to distinguish between today is oxidative phosphorylation and chemiosmosis. Two important steps. So,
Jerry?
yes?
When you do the, oh, okay, I'm drawing a blank, but the inside of the mitochondria, the cytoplasm, right?
Okay, this is the matrix.
The matrix,
of the mitochondria.
Okay, so you put all the minuses there. So
yes.
Those are the OH?
They are OHs. They could be other negatively charged ions.
So just...
Again, but picture it. This is a phospholipid bilayer, isn't it?
Right.
And that phospholipid bilayer, if you look at this, remember this is the phosphate-containing group
right,
on
so
glycerol, the fatty acids.
side.
The phosphate group is negatively charged. So every phosphate group is negatively charged. So this entire membrane potentially is negatively charged. Now what happens is, if you put a bunch of protons over here, they've neutralized the negative charge. That's why it's plus. If I take the protons away, both sides are going to look negative at the membrane there.
Got it.
Now there's going to be other sodium ions positively charged that would neutralize that, but right at the membrane it's going to be that way. Okay, so let me give you some numbers which I think are pretty interesting. First of all, we're going to get in this process 36 ATP per glucose in complete aerobic respiration of glucose. (36 ATP per glucose when we have complete aerobic respiration of glucose. Now, how much ATP do we use in a day? We're going to see that ATP and ADP move through these cycles in impressive quantities. For example, if you folks have a very relaxing, kickback sort of couch potato day, maybe all you're going to do is eat potato chips and watch the Super Bowl, something like that, you would use an amount of ATP equivalent to half your body weight. If you were out, so imagine you weigh 160, there's 80 pounds of ATP in a day. But if you were active and you were out cutting and splitting, say, firewood and hauling it, you might use 2 to 3 times your body weight in ATP in a 24-hour period. So if you weigh 160 pounds, we're talking 320 pounds of ATP in a day. Now, how many pounds of ATP do you have in your body right this minute? A fraction of a pound. So what this means is this stuff is cycling through very, very frequently and in large quantities in a particular day. All right, I promised you a big, bad definition of oxidative phosphorylation. Here it comes. This is going to tell the story of how we get so much energy from our ATP. So oxidative phosphorylation is going to consist of 3 parts that we want to talk about. We need to have, first, a series of enzyme-catalyzed reactions. In this case, that's going to be glycolysis and the Krebs cycle. A series of enzyme-catalyzed reactions that provide a source of high-energy electrons. So glycolysis and the Krebs cycle provide us with a source of high-energy electrons, right? And where are these electrons found? Well, they're found in NADH and FADH2. Those are the coenzymes which temporarily store these high-energy electrons. Glycolysis, pre-Krebs, and Krebs are the reactions that we're talking about here. Now, the second part of oxidative phosphorylation will involve an electron transport system. Which happens to be embedded in a membrane. An electron transport system embedded in a membrane. My picture in the upper right-hand corner, right? The ETC that's embedded in the membrane, that's what we're talking about here. And this electron transport system is going to uses the energy in the electrons to create a proton gradient. To create a proton gradient.